Bismillahirrahmanirrahim. Assalamu alaikum, my dear students. In previous video clip, we discussed meiosis, right? Who discovered it, in which organism it was first found, right? And then its types. Then I told you that it is completed in two phases: meiosis first and meiosis second. Now let us see how this all goes on. This meiosis first, it includes, as we saw in mitosis, the karyokinesis, which is followed by cytokinesis. So here the cell undergoes karyokinesis, that is new division of nucleus. But since the meiosis is completed in two divisions, first and second, we write here karyokinesis first. And this karyokinesis, similar way as it is completed in mitosis, is completed in four phases, right? And those four phases are the prophase, right, the metaphase, anaphase, and telophase, which is then followed by this cytokinesis. And since it is the first meiotic division, these are marked by first, prophase first, metaphase first, anaphase first, and telophase first, right? But here the prophase first takes longer time, though it is completed in short period of time, but it has different stages which we have to, and it is completed in this prophase is completed in 5 stages. It is completed in 5 stages. This is what I am going to give you the introduction of this meiosis, right? The first is it is leptotin, right? Leptotin. Then next stage, that is zygotin, which is followed by next stage, that is pacitin. It is followed by the fourth stage, which is diplotin, and diplotin is then followed by dikinesis, and it completes the prophase first, which is then followed by metaphase first. Metaphase first is followed by anaphase first, and anaphase first is followed by telophase first, which we have seen in this mitosis. But here the difference is that this division is reductional division. Here the chromosome number in these daughter cells gets halved. That is, what is the number in the parent cell, the daughter cells have the half the number of chromosomes as compared to that, right? That is, a 2n cell gives rise to 2 daughter cells which are n, having n number of chromosomes, right? That is, a diploid cell gets divided into 2 cells which are haploid, having the half number of chromosomes compared to this parent cell, right? These all stages after karyokinesis, then comes cytokinesis, which involves the division of cytoplasm, and as such 2 cells are formed having the half number of chromosomes. Since it is meiosis first, so we mark it as cytokinesis first. See, when we say this meiosis, the cell, which is basically 2n, which enters this meiosis, first undergoes interphase, where it prepares itself for the cell division, as we have seen in cell cycle in mitosis, right? But here, when these in cytokinesis, when these 2 haploid cells are formed, they now undergo the second meiotic division. Here they undergo the second meiotic division. But before that, for a short period, they go under a preparatory phase. That is, these 2 cells prepare themselves for meiotic second division. That is, these 2 haploid cells prepare themselves for meiotic second डिवीजन, राइट। और इस फेज को जो है कहते हैं इंटरकाइनेसिस, नॉट इंटरफेज, याद रखें, वहाँ पे इंटरफेज है, ये इंटरकाइनेसिस। एंड हाउ इज इंटरकाइनेसिस डिफरेंट फ्रॉम इंटरफेज? दिस लैक्स एस फेज, इसमें एस फेज नहीं होता है, दैट इज डीएनए रिप्लिकेशन नहीं होती, राइट। इसके बाद जो है ये इंटर होती है मियोटिक सेकंड, ये जो मियोसिस सेकंड है इसमें, और इन मियोसिस सेकंड अगेन these 2 cells, right, the haploid cells, right, which are formed in meiosis first, they undergo first karyokinesis, and we mark it as karyokinesis second since it is meiosis second. Then, and this karyokinesis again is completed in 4 phases, that is prophase, ठीक है ना, metaphase, anaphase and telophase, right? But we mark them as prophase second, metaphase second, anaphase second and telophase second. It is then followed by cytokinesis. Here we say it is cytokinesis first, right, and here it is cytokinesis second, and as a result 4 cells are formed from these 2 cells, right. So in meiosis, first a 2n cell, right, it undergoes first meiotic division, right, first meiotic division, and forms 2 cells, 2 daughter cells, which are having half the number of chromosomes, right, and these 2 cells undergo, right, second meiotic division. They undergo second meiotic division, right, which is actually equational division. ये फर्स्ट meiotic division जो है ये है इट इज रिडक्शनल, जैसे आपने देखा कि 2n था यहाँ पे, तो ये जो हैप्लोइड डॉटर सेल्स भी नहीं, ये हैप्लॉइड है हैविंग द हाफ नंबर ऑफ क्रोमोसोम, दैट इज द क्रोमोसोम नंबर इज रिड्यूस्ड, दैट इज व्हाई इट इज कॉल्ड एज रिडक्शनल डिवीजन। तो यहाँ पे जो है ये इक्वेशनल डिवीजन है और सेम नंबर ऑफ क्रोमोसोम रहता है इन द डॉटर सेल्स, जो n number of chromosomes, and as such, ठीक, 4 haploid cells are formed from a parent diploid cell और इन ही जो ये सेल्स हैं इनको हम कहते हैं गैमीट्स, राइट। तो आगे जो है विल डिस्कस इन डिटेल दीज स्टेप्स, राइट। दिस लेप्टोटीन, जाइगोटीन, पैकिटीन वगैरह कैसे होता है मियोसिस फर्स्ट और मियोसिस सेकंड, एंड दैट विल बी इन द नेक्स्ट वीडियो। अस्सलाम वालेकुम।